So hello everyone and welcome to another Geomicrobiology Network seminar series. My name is Karen Cameron and I am delighted that we have Liz Bagshaw joining us today. Liz is a senior lecturer in biogeochemistry and glaciology at Cardiff University. And I have to say that of all the people I know working in the microbiology of the terrestrial cryosphere, Liz clearly stands out from the crowds owing to the innovations and the technologies that she's brought to the field. And these dimensions of her research have brought about transformative knowledge gains and also a lot of opportunities for glacial research moving forward. So I've always been really intrigued to read about Liz's latest ventures, and yeah, I'm definitely looking forward to hearing her presentation. And so Liz has worked on both the supraglacial and the subglacial environments, and she's worked on glaciers from the Arctic to Antarctica. As I say, I'm truly delighted that Liz is joining with us today and giving up her time. I know she's super busy. In these sessions, we really welcome interactivity, as I've said before. So after Liz finishes her presentation, if you have comments for her or if you have questions, then we really encourage you to turn on your mics and engage with Liz and even engage with each other if possible. You're also really welcome to write any questions that you have in the chat, and I can try and ask those of Liz at the end. So this session will be recorded, so keep the language good and nice. And yeah, I'll hand over to you now, Liz, and yeah, feel free to start whenever you feel ready.
Great, thank you for that really kind introduction. Yeah, it's nice that yeah, Caroline, I don't know where Caroline and I met, but we definitely met randomly on a train in Copenhagen once. So our paths have crossed many times over the years. So I was delighted that she invited me to do this seminar, and I'm excited to see you all in the audience. So I'm going to attempt to share my screen right now. Can you all see that? Yep, great. Okay, so this talk is kind of a little bit of a retrospective for me because my main research that I do these days isn't so much on the ground with cryoconite, but my PhD students do still do a lot of work in cryoconite. So I decided to present kind of an overview of what I've done on cryoconite so far and what my students are doing now, and as Karen said, explore some of the new techniques that we've been using to try and understand these really fascinating systems. So even though I'm the one giving the talk, really much of the work has been accomplished by my students, Ewa Poniecka and Jazz Miller, and also with support of my supervisors from the past, so Martin Tranter, Andrew Fountain, and my colleagues today, Henrik, Anne, Arwin, Amelia, and my postdoc, Mike Pryor-Jones, and the hundreds of people who have helped us out in the field over the past few years. So I wanted to do a kind of what, where, why, and how, mostly because I'm not very good at doing punny titles. Oh, there we go. I might have just clicked 6 slides forward by accident. No, it's all right. So the reason that cryoconite holes are interesting is because they're a key player in the glacial biome. And until about sort of 20 years ago, people thought, nah, glaciers far too cold, dark for half the year, can't be anything living there. But as we've seen, and as you all know in this seminar, that the glacial environment is actually a surprisingly hospitable home for microbial communities, and the cryoconite holes up here are a really key player in the glacier biome. So cryoconite holes, for anyone who's not interested, the thing that I loved about preparing this seminar was usually my first slide is let's define a cryoconite hole, let's introduce everyone to it. But I found that this week there was actually an article about cryoconite holes in the Guardian newspaper. And when I started doing a little news search, not only was this cool one about tardigrades in Arctic cryoconite hole, but Arwin's group has had some lovely press in the Guardian over the few years. The Black and Bloom project has had a whole series on the BBC. So suddenly I don't need to start with a massive introduction to what a cryoconite hole is. But I still will, because I think it's really important for us to get our definitions right when we're talking about these systems. So the superglacial environment, so this is a sort of aerial view of the superglacial environment, a lovely piece of artwork that was out in Science a few years ago. And you can see the cryoconite holes in the middle there. These, I love that their dust, soot and microbes form black gunk that coagulates in pits. That might be one of my favorite definitions of cryoconite. But what you can see is cryoconite is part of a whole superglacial system, all of which are linked together by meltwater at various points in an annual, seasonal, or even multi-annual cycle. And the stuff that happens in cryoconite holes can impact the rest of the superglacial environment, which in turn can impact downstream systems. So cryoconite forms from debris that's blown up onto the surface of the ice. And because that debris tends to be dark coloured, it absorbs more solar radiation than the ice surrounding it and melts down into the ice, forming a lovely little pocket of liquid water, including sediment and microorganisms that are transported with the windblown debris. And because you've got this little pocket of liquid water ranging from a few centimetres to up to a metre, maybe two, I'm not sure quite when it stops being a cryoconite hole and becomes a cryolake or a pond or whatever, but let's go with a few centimetres to a few metres at most. You get this little mini laboratory where you can study processes on a small scale, knowing all the inputs and outputs. So as a biogeochemist, I love the cryoconite system because it's at a manageable scale to figure out who's doing what in the ecosystem and what the inputs and outputs are. So within the cryoconite, you've got this whole mix of sediment, microorganisms and other kind of biological material. They tend to be seeded from the surrounding environment, so there's not usually anything particularly exotic in there. But what you do have in a cryoconite hole are the survivors that are transported from the surrounding environment. So you've got a whole bunch of cyanos, you've got chlorophytes, you've got rotifers, some tardigrades, although according to that Guardian article, not so many tardigrades around. There was an interesting paper by the Polish group, Krzysztof Zawierusza, sorry if I mispronounce his name, talking about the tardigrade conundrum, no, sorry, the nematode conundrum recently. But basically you have a whole bunch of stuff in the cryoconite holes. The majority is made up of heterotrophic bacteria, but you do have autotrophs in there. And we're interested in studying these microorganisms because they're these little mini laboratory that exists on its own, potentially isolated from the surrounding environment. They're a really good model for life on other extraterrestrial icy bodies or for how life might have persisted through Snowball Earth. The microbes within the holes have to withstand extremes of temperature, they have to withstand freeze-thaw, and they have to withstand extremes of UV exposure, including 6 months of dark, well, depending on where you are on the globe. So they're really, really fascinating. The holes themselves, because they contain all of the different parts of the community, you have a complete food web within that cryoconite hole. This was a cool diagram prepared by Chris Bellis a few years ago for a book chapter we did. And you can see here that you've got the primary producers, you've got the decomposers, you also have some grazers, and there's even viruses active in cryoconite holes. So because you've got all these different parts of the food web, you have a complete self-sustaining system. It doesn't really need many inputs from the surrounding environment in order to function. You find cryoconite holes basically wherever there's a glacier and some dirt around, there's the potential for cryoconite holes to form. And this is that paper by Krzysztof Zawierusza that was published recently. And they did a really cool survey of the macrofauna in the holes. But what I like about this map is you can see the huge spread of cryoconite hole research across the globe. Basically, where there's glaciers and dirt, people might be studying the cryoconite on it. The majority of my work's taking place in the dry valleys in Antarctica, in Greenland, and a little bit in Svalbard and a little bit in the Alps. So most of the stuff that I'll be presenting will be from a sample suite from those locations. But as you can see from this map, there's cryoconite all over the place. The interesting thing about the global distribution of the cryoconite is the physical conditions control what's going on in the holes themselves. So depending on the ice temperature and depending on the solar radiation patterns, that controls how exposed the cryoconite is to the surrounding environment. And generally, the holes in the Antarctic tend to retain ice lids throughout the melt season. So you get in this diagram here, I'm just checking, you can't see my little zoom sidebar on your screen, can you? No, okay, phew, just checking because half of my screen's covered up with your lovely faces. So what this diagram shows is the development of an ice-lidded cryoconite hole. So your sediment falls onto the glacier, starts to melt down, and because the ice is so cold, the ice refreezes back over the top of that melted debris. The ice that refreezes over the top is very transparent, it's beautiful, clear ice. So you end up with ice that light can very, very easily penetrate through. That continues to heat up that debris at the bottom, and that retains a little layer of liquid water. But that refrozen ice lid stays there throughout the season. And some of the research that that Martin Tranter and myself did in the Antarctic dry valleys, we looked at trying to figure out how long some of these holes have been isolated from the surrounding hydrological system. We were finding holes that have been isolated for up to kind of a decade at a time. But because you've got that complete food web and you've got that mineral debris in the cryoconite itself, you've basically got this self-sustaining community. So it doesn't matter if they have no hydrological inputs, they've got everything they need from chemical weathering going on the cryoconite and from a very, very small amount of ice melt around the sediment. And this lovely image from Mark Stiebel shows all the different components, and all those forms of nutrients are just swapped around the different components of the ecosystem. So they just carry on. The meltwater resupplies just enough. And then when these holes are hydrologically connected to the surrounding superglacial drainage system, all that kind of good stuff in the meltwater gets flushed out and has the potential to fertilize downstream ecosystems. But we'll talk about that a little bit more later. So what are the key differences in the communities between the Arctic and the Antarctic? So this is some analysis that my PhD student Jazz has been doing, looking at a whole suite of samples from across the Antarctic, from dry valleys, from right into the Utstein and Scoop, and then across the Arctic from Greenland, Svalbard, a few other places that we'll see on the next slide. And what Jazz has found is that there is a real clear difference between the communities in the Arctic and the Antarctic. And this is the kind of composition of the prokaryotes. Please don't ask me any difficult microbiology questions. I might have to phone Jazz to help me out. I'm really a biogeochemist, despite this being a geomicrobiology seminar. But what you can see here is that there are distinct differences between the Antarctic samples down at this end and the Arctic samples. Jazz got a paper coming out soon, so you can read all about these in more detail and ask them all the difficult questions. So what I'm really interested in is trying to figure out what is it that's controlling the difference between these communities. Is it that the community is different because of where it was seeded from, or is it that the physical conditions control what can actually survive in the holes? And if we look at these photographs on the right-hand side of your screen, so the top three, A, B, and C, these are Arctic cryoconite holes. So these are some of Ewa Poniecka's pictures. And what you can see there is these holes remain open to the atmosphere during the summer melt season. They might have a little bit of a thin ice lid. So you can see on image B, there's a little bit of an ice lid there. But image A and image C are the kind of classic Arctic cryoconite hole, whereas the lower images, D to F, these are the ones that refreeze those ice lids over the top. And generally in the colder ice in the Antarctic, because you're right down with your solar angles spread right out over a wide area, and you've got much lower temperatures, those ice lids are retained. But because the sun can penetrate through those lovely clear ice lids, you do still get photosynthetic activity going on underneath the ice, even though it's sealed off from the atmosphere. So what Ewa Poniecka has been doing is trying to figure out which of these microorganisms can survive. She was exploring samples from Antarctica, Greenland, and Svalbard, and trying to see which ones were the hardiest ones, and trying to understand if it's that the Antarctic species are more badass than the Arctic species. And so Ewa basically tortured a load of isolates from some of her incubations in the lab. And what Ewa found was that many of the isolates can easily survive multiple freeze-thaw cycles. She nuked them with over 100 freeze-thaw cycles, and some of them were absolutely fine, still viable. Interestingly though, some of them did lose viability after just a few freeze-thaw cycles, and there didn't seem to be a clear pattern on which ones survived and which ones didn't. And this paper's just come out in Frontiers in Microbiology, so do have a read of that. And there's a really nice special issue in Frontiers in Microbiology that's just come out, which has got lots of cryoconite stuff. So I'd recommend going and having a look there. But you can see which ones lost viability if you read the paper in detail. She also looked at which ones were happiest at what temperature. And again, they all seem to cope pretty well. Once you get up to about 30 degrees, then the cell counts begin to drop. But generally, they're all pretty fine, no matter what temperature you expose them to. The same with the salinity, the plot in the top right here. So the percentage of isolates growing does begin to drop as the salinity increases. But I mean, the salinities that she was testing were really far beyond anything that we would experience in the freshwater cryoconite environment. So it was really surprising how much they can tolerate. And then the bottom right-hand plot is the pH of growth media. So Ewa tested pHs right from I think 3.5 all the way up to 11, and samples pretty much survived whatever she threw at them. You can see that the Antarctic samples, the medium pH, the ones that with the highest viability, had higher pH than the Greenland and Svalbard samples. We think this is because the pH of the Antarctic holes does seem to be much higher than the Greenland and Svalbard ones. Because that ice lid remains in place, it means that after photosynthesis occurs, they can't degas. And Martin Tranter wrote a great paper about this back in 2004, which I'd recommend a read of. That lack of contact with the atmosphere just drives up the pH. So when we do pop the lids of some of these sealed off Antarctic holes, we see pHs up to 9, 10, 11, so they can be really high. So it wasn't surprising that the pH of the Antarctic samples, the tolerance of the Antarctic samples was much higher. But the conclusion of these experiments was really that whatever you throw at them, the microbes survive. So I kind of touched on this a little bit earlier on. Now we know kind of what cryoconite is and where it is. Why are we so interested in it? The first is because we love them as a model for how life persists in extreme environments. And there's a lot of work going on Cryoconite as a model for life on icy extraterrestrial moons. I'm really interested in the moment on how life persisted through Snowball Earth. And this was kind of kicked off by a paper written by Paul Hoffman, Mr. Snowball Earth, in 2016, who looked at, he called them cryoconite pans. I don't know what a cryoconite pan is. He made up a new thing. But I think this basically means a large dump of cryoconite. So the photograph in the bottom right here is from Canada Glacier in the McMurdo Dry Valleys, where many of my samples are from. And you get kind of these depressions on the ice surface where you get big accumulations of cryoconite. We refer to them as sort of cryo lake systems, where the meltwater's flushed down a bunch of sediment and it's ended up accumulating in a hollow on the glacier surface. So this could be a kind of model for what he means by a cryoconite pan. Could also be something in that you tend to see more on ice shelf systems. This image in the top right, this is from Ian Hoare's paper on the McMurdo ice shelf, which has some really beautiful algal mats and some of these big old piles of dirt. And what Hoffman is arguing is that these are how life could have persisted through the Snowball Earth system. But what we don't really know is how realistic that is. So Jazz Miller's PhD project is to try and understand what's going on in the cryoconite and figure out who's there and compare that with what might have been around through Snowball Earth. So again, no difficult questions about the microbiology here, but this is Jazz's assessment of the Antarctic and the Greenland samples for the Eukaryotes. And what Jazz has then done is gone through the fossil record and tried to figure out which of the organisms that we identify in modern day cryoconite are also written about in the fossil record, or there's some evidence in the literature for them having persisted through Snowball Earth. And you can see from this big old list of green ticks that a whole bunch of stuff that unknown cryogenian organisms are present in the modern day cryoconite. So Jazz's research is ongoing. They're doing some really cool incubations to try and mimic Snowball Earth conditions in the laboratory, just on the small scale. Our lab's not that cold, but trying to figure out how things could have persisted in these cryoconite pans through Snowball Earth-like conditions. But the thing that I find really fascinating about cryoconite hulls is how they kind of do this sort of swapping around of nutrients within the ecosystem without having any external inputs. And this image borrowed from Joe Cook down the left is a really nice illustration of kind of how the cryoconite hulls contribute to the whole supraglacial system. And what you see, what's critical in this image is that little meltwater stream trundling down the glacier. Everything that happens in the cryoconite hulls is then flushed into these meltwater streams and ends up either going over the rest of the supraglacial system, or ending up going down a moulin into the subglacial system, or even in the Antarctic, where you have limited subglacial drainage in some of the Antarctic glaciers, that goes straight into the proglacial systems. And so some of the work I was doing in the dry valleys was trying to figure out what contribution the cryoconite had to the wider ecosystem. And there's been lots of work done trying to quantify how carbon fixed in cryoconite hulls supports the rest of the supraglacial system. So this has been kind of something rumbling on in the literature probably over a decade, trying to figure out what is the actual contribution of cryoconite to carbon accumulation on the ice surface, and then how does that influence the subglacial community and the downstream systems that rely on that glacial carbon. And there's been lots of great work done on the carbon export from glacial systems. Gemma Wadham had a review paper out in, I think, Nature Communications last year that's really worth a read to kind of get the overall picture. And I think Marek Stiebel's group's been doing lots of stuff on the carbon export. Karen's been doing some stuff on that. So, you know, I'm not going to try and cover the whole carbon story. What I want to talk about here is the kind of nitty gritty. How does this carbon actually accumulate? How much is there? And does the structure of the cryoconite influence how much carbon might be being added to the system? So what myself and Ewa Poniecka have been doing for a number of years is looking at these different kind of cryoconite structures and figuring out how they accumulate carbon. So if we look at these three images here, on the left there's a cryoconite granule, which I'll talk about in a minute, then a sort of more classic deep cryoconite hole, loads of black gunk with a nice thick water layer, and then on the right just a kind of sludgy mess where most of the water's drained off. And do these different structures have different carbon accumulation sort of processes? The problem is, how on earth do you measure that? So this is where I had to start thinking a little bit more creatively, and going back to probably my very first postdoc after my PhD, looking at what kind of technologies can be used to assess stuff occurring in the field. So a lot of the cryoconite research is necessarily done back in the lab. We bring samples home, then we mess with them and measure them in a nice, easy environment. But once you remove the cryoconite from the system, particularly in some of these isolated holes in the Antarctic, actually you're changing how the system works. So what I really wanted to know in some of my postdoc work was, can we measure some of these systems in situ, and can we get meaningful data on what they're doing on a day-to-day basis? So I started exploring methods for measuring some of the stuff we were interested in, and you can get great sensors for oxygen, which measure the dissolved oxygen concentration in water using fiber optic technology. So this is the bottom left. You can see a little silver probe about the size of a pen. Encased in that silver probe is an optical fiber, and then that works with an excitation system, and on the end of the optical fiber is a sensing layer made of oxygen sensitive fluorophores. So if you could get this to work in a cryoconite hole, then you could kind of observe what the microbial community is doing sort of as a whole. Is there oxygen production peaks when it's favorable productivity conditions? Can you use oxygen to figure out when the carbon's being accumulated? This sounded straightforward. Yeah, I'll just buy an instrument. I'll just take it to Antarctica, it'll be fine. But anyone who's worked with technology will know that that's really not what happens. We had to do lots of modifications. We spent a long time figuring out which versions of these sensors would actually work in the cold, which ones survived those freeze-thaw cycles. We found one where the sensor itself was absolutely fine in the cold, but the electronics really didn't like the cold. So you can see in this picture, the oxygen meter is wrapped in insulating foam. I also took it a hot water bottle every other day. So technology is, I thought this would be the end of my my days of washing bottles, and I'd be like, oh, working with technology is so much better than working with geochemistry. Apparently it's not. There's a whole host of other heartbreak. But what you see on the right is some in situ data that we managed to get from some probes that we froze into some isolated cryoconite holes. And generally we found that the oxygen levels stay fairly undersaturated, but you do get these cool little peaks happening, probably due to freeze-thaw processes releasing stuff from the ice below. So that was exciting. But we weren't really able to get much about the community activity because we were just working in the water above. So we brought a load of samples home and tried to run them in a very long-term incubation under Antarctic conditions in the lab. So we controlled the light, we controlled the temperature, and then every few hours, then every few days, measured the oxygen balance in the water above the cryoconite sediment. So this kind of incubation technique has been sort of pioneered probably by Alex Nisio, brought the technique to the field in the late 2000s. Mostly looking at 24-hour incubations, which is kind of the biology standard for biological oxygen demand sort of processes. What we found with the cryoconite is a 24-hour incubation, particularly with the Antarctic ones, isn't really sufficient, because once you disturb your sediment when you put them in your incubation vessels, you mess with the layering of the community, and they don't really do anything in 24 hours. And what we found in the long-term incubations is what you can see in these plots is sort of the first like 20 days or so, it's all just a jumbled mess. And then after that, the light bottles and the dark bottles begin to diverge, and you do find that you get net ecosystem production. So you get an accumulation of carbon over time if you leave your incubations long enough. So that was cool in the lab. We then tried to figure out, right, what are the processes that are going on? And we kind of came up with this grand conceptual diagram, figuring out, okay, if we've got an oversaturation of oxygen in the water layer, then what must be going on in the sediment in order to account for that water layer? And this came out in Geochemical Perspective Letters in 2016. And we kind of came up with this theory about sort of diffusion from the heterotrophic activity and also from carbonate dissolution in the cryoconite layer. And then that pops up into the water and you end up with net production. But this can only happen if oxygen is depleted very, very rapidly in the sediment. So if you look at picture F in that little schematic, you can see that the oxygen decreases very, very, very rapidly in the sediment. This was our hypothetical model of what must be going on. But then I thought, well, we've got to be able to measure that. So I started exploring some of the different formats of the fiber optic systems. Rather than a larger probe, you can actually get a needle tip one, which you can use to insert in the sediment layers and do some very, very fine profiling of what's going on in the sediment. So my first sediment profile was this one in the top right-hand side, where we looked at the difference between the water and the sediment layer. And lo and behold, you do get low oxygen conditions in the sediment layer. So at this point, I recruited Ewa Poniecka and set her to work trying to figure out where those anoxic layers start and what role does the anoxic part of the community play in the carbon accumulation as a whole. So Ewa took one of these microoptodes out onto the Greenland ice sheet to try and do a whole bunch of profiling. And Eva's just submitted her thesis, and she found that the anoxic microbes are a significant part of the community. We find loads of them where you've got some kind of superglacial input to the system, but even when you're far away from the subglacial part of the system, but even when you're far away from that, right in the center of the ice sheet, you do still get anoxic zones in the debris. And you can see this nice little image that she's made down on the right-hand side showing kind of how the oxygen profile occurs in the cryoconite hull. And she's done a whole bunch of experiments with this, the anoxic part of the community, which you'll be able to read about in some papers soon. So Ewa kind of trundled around with her microsensors, looking at how the structure of the cryoconite affects the anoxic layer. So the top three plots here are oxygen profiles in different kinds of cryoconite. And basically where the sediment is thicker and more of a clump, you get those anoxic zones are more prevalent. If you take that sediment and you disturb it and you start a new incubation, so you oxygenate the sediment, mix it all up, let it get to air saturation, those anoxic zones establish really, really quickly once you've moved that sediment again. So what you can see in the bottom series of plots here is the oxygen profile taken over a couple of hours after disturbing the sediment. And you can see that that anoxic zone very, very quickly develops. Within a couple of hours, you've got a full anoxic zone really close to the surface in the sediment. So the next thing that that we wanted to figure out was, okay, what does that mean for the whole ecosystem? We know that these anoxic zones are there. What does that mean for the ecosystem production? So again, we did some of these light dark biological oxygen demand bottle incubations. Eva did some of those in the field in Greenland with the Black and Bloom project, and compared different kinds of samples, incubated them on the ice surface, and looked at the net ecosystem production. So this NEP is how much carbon is being produced by these little communities. Then, because we don't get very long in the field, she wanted to see if we get stuff happening if you incubate the sediment for much longer. So she brought some of those Greenland samples home and incubated them for a long time, and also did the same with the Antarctic samples. And you see here that you do get a difference between the Greenland and the Antarctic ones. Greenland ones seem to be more productive, but in all of them, you are getting anoxic zones within the sediment, and it happens pretty quickly. So all the kind of little incubation works that we've done have shown that really, if you want to mimic those field conditions, you've either got to measure them in the field, or you've got to do long-term experiments. So you've got to give them time after you disturb them. We love using oxygen optodes. They're pretty handy for assessing community activity. I find them much easier than messing about with radio labeling stuff. And Jazz has done some side-by-side comparisons, which hopefully they'll be able to publish at some point. And we find that, yeah, it's a good enough assessment. You might not get the precision that you get with radio labeling, but if you want to know the community activity, then you can find out. Once you move those sediments, production kicks off really, really rapidly once you add water to them again. And even after frozen storage, we've had samples in the freezer for a decade, and then we pull them out, add some water, and they just get going really, really quickly. And we find that generally there's a sort of phototrophic activity dominates at the start of these systems, and then heterotrophic activity kicks in as those as the community sort of self-organizes itself into its layers. And it looks like the peak of activity occurs a little bit sooner in the Antarctic experiments rather than the Greenland ones. This could be a reflection of the Antarctic growing season being a little bit shorter, but this is only done from a couple of samples, so I don't want to make too many sweeping statements on that. The other thing that we've been exploring is the difference between the structure of the Arctic versus the Antarctic systems. And one of the kind of classic features of cryoconite in the Arctic is that they form these little things called granules. And these are little clusters of minerals and microbes that are all stuck together with EPS, extrapolymeric substances. Whereas in the Antarctic, you don't tend to see these granules forming. And I was really, having kind of done the majority of my work on the Antarctic ones, I just couldn't really figure out what the difference was. And so we've been exploring some of the what's going on in the Arctic system versus the Antarctic system. And the key difference is the presence of this ice lid in the Antarctic. And what this plot on the right shows is the difference between the photosynthetically available radiation received on the ice surface versus that received at the bottom of the cryoconite hole. And you can see that that ice lid provides a really, really good filter for radiation coming into the hole. So the Antarctic cryoconite is actually pretty well protected from extremes of solar radiation. The Arctic cryoconite generally doesn't have that ice lid during the growing season. So they've got to find other ways of protecting themselves. So I think one of the reasons for this granule formation is as a photoprotection strategy. So the phototrophs kind of cluster around the edge, and then the heterotrophs stick in the middle, and you end up with a kind of mobile community. These granules can roll around. So as they roll around, that means that everyone takes a turn on the top, and so you get some kind of photoprotection. It also means that you've got a sort of Meals on Wheels service. I'm just going to skip forward here, because if you look at these beautiful images from one of Birgit Sattler's papers, these like cyanos kind of stick everything together. And so that means that you're keeping your energy and water sources close. And going back to this image that Marek made of a cryoconite hole, what I think is going on in the granule is the stable system. So in the Antarctic, as I said, you don't get hydrological connection very often. These holes can be isolated for up to a decade. So anything that's within the hole is accessible to the whole community until it's flushed with meltwater, but that doesn't happen very often. In the Arctic, the cryoconite are flushed very, very often. So if you want a stable system, you need to figure out a way to kind of stick your food to yourself. So I think the granules basically operate as a sort of Meals on Wheels service for the community there. So not only does it provide photoprotection, but it means it keeps all their energy sources really in close proximity. So I'm just going to finish with a little bit of a taster in my other work. Karen asked me to talk a bit about the tech development that I'm doing for the subglacial system. So we've been talking a lot about the cryoconite on the surface. What happens to that meltwater when it goes down the moulins, when it ends up in the subglacial system? If you think right back to the first image that I showed of the glacial biome, one of the biggest areas for microorganisms to colonize is the subglacial system. So I'm really interested as a biogeochemist in what's going on in that subglacial system. Problem is, it's underneath a couple of kilometers of ice, so it's a real pain to observe. So I've got some funding from the Engineering and Physical Sciences Research Council to develop a meltwater monitoring probe that goes underneath the ice and returns data wirelessly to the surface. And what Cryoegg is, it's something that I started working on with Gemma Wadham at the University of Bristol, and have since brought it to Cardiff and got a postdoc, Mike Pryor-Jones, working on the project. It's a spherical probe about the size of a grapefruit, which contains a suite of pretty simple sensors and a radio transmitter. And the purpose of Cryoegg is for us to be able to send it underneath the glacier and measure some of those characteristics of meltwater and transmit those data wirelessly up to the surface. And that's handy because trying to deploy a probe with on a cable is really, really hard in fast-moving ice. Even if you get a borehole drilled to the bed, as soon as that ice moves, your borehole stretches, your cable ends up severing, and then you lose all your data. With the Cryoegg, the idea is that you deploy it, and then you just have a receiver on the surface. At the moment, we've got quite simple sensors in there, but the measurements can be quite simple if you can get a long time series and a good spatial resolution. So the Cryoegg, you can see that in that picture, Mike's holding it there in a little net. It's about to be deployed into a borehole on the Swiss glacier. Inside Cryoegg, there's a few circuit boards, there's a radio transmitter, and you can see the little antenna sticking out the front. And in the middle picture there, B, this is our antenna setup. So this is at the East Greenland ice core, which is being drilled up in a fast-flowing ice stream in the northeast of Greenland, in the Negus ice stream. And what you can see in that image there, the green plastic frame is actually just holding up an antenna. It's actually a kid's climbing frame that we use to deploy our antenna, because the antenna needs to point downwards, because at that point the egg is underneath 1.5 kilometers of ice, and it's sending its data back in real time to that antenna, which is then being recorded on the laptop. So we've been trying to develop this technology for a number of years, and we've had some moderate success in the past couple of years. In the East Greenland borehole, we managed to get live data transmitted from 1.4 kilometers beneath the ice. We get a live feed of the pressure and the temperature, and also the electrical conductivity, but the conductivity in the borehole is 0 because it's just drill fluid at this moment. But in the future, we intend to deploy it underneath the ice to figure out what's going on with the geochemistry of the subglacial system. But the most exciting thing to take away from this plot is that all this data was transmitted wirelessly through 1.5 kilometers of ice. We also use the egg for some in situ salt tracing underneath the glacier. So this is a way of estimating the discharge of subglacial drainage systems. So figuring out how much water is flowing around underneath the glacier. So we did this at the Rhone Glacier in Switzerland, and we got a lovely salt tracing peak, and we were able to figure out the volume of water in our moulin. So the next steps for the Cryoegg, we're hoping to deploy down to the bed of the East Greenland ice stream at 2.5 kilometers, and then eventually release it into the subglacial system. Then hopefully we'll be able to install some of the sensors that I've been talking about. So the oxygen sensor, for example, I want to install that on the Cryoegg, and hopefully follow it with some pH sensors too. So my conclusion, Cryoconite holes are really freaking cool, in my opinion. And they host some really resilient and productive microbial communities. But if you want to observe them, you need to use some novel techniques, and you need patience as well. You can't, they take their time to get going. It looks like the community behaviour is influenced by the physical structure, but we think they're a nice candidate for refuges for life during Snowball Earth conditions. And with that, I will thank you all for your attention.
Thanks, Liz. That's great. If anyone has questions to get going with, then please shout out or raise a blue hand, or however you like to do it. And there's also the opportunity to use the chat function in Zoom to write any questions that you have. So if anyone wants to get started.
I've got a question.
Go for it.
Hi, Liz.
Hello.
Nice that the Cryoconite is still there. Ari, the Meals on Wheels idea, do you think that motion of the Cryoconite granules, how do you think that's being generated? Is it sort of abiotic factors, like sort of movement on the surface of the Cryoconite hole, energy that's being transferred down, or do you think it's sort of cyanobacterial motility that's driving that movement? Any ideas about what's going on?
I think the motion of the granules themselves is within the meltwater, because you even within a Cryoconite hole, if you sit and stare at a Cryoconite hole, which I certainly have spent many hours just watching them, you see that you get kind of little convective currents even in a seemingly disconnected Cryoconite hole. So there's always motion in the hole, and then once you get some of these superglacial melt streams connecting into the Cryoconite hole, you see the granules floating down, well, not floating, they're kind of saltating down the bottom of the superglacial streams. So I think the granules themselves are moved by abiotic factors, but within the granule, it might be that cyanobacteria motility, like they can move up and down according to where they are situated in the granule. Like maybe they all take a turn to go on the outside, and then they have a little bit of a rest. But yeah, that would be more your department than mine.
I think James has a question.
Hi, Liz.
Hello.
How are you? I was going to ask about your productivity measurements. It seems to be that you're measuring everything per litre of Cryoconite water, I guess. To what extent is it impacted by how much, grams of sediment there would be? And then if you were going to upscale that, how do you think about fluxes across the ice sheet surface if you're thinking in litres of meltwater and grams of sediment, and how that's just distributed?
We tend to do it all in grams of sediment because that's kind of easier to manage. So I mean, that's one of the difficulties in doing it, trying to do it in situ in a cryoconite hole, is you don't know your grams of sediment, so you can't get your traditional productivity measurement. If you do it in situ in a hole where you haven't weighed out your sediment, then yeah, you can just get a kind of oxygen's high versus oxygen low. But for the kind of when you want the detailed community productivity, then yeah, I've been doing it. We've been weighing it and as grams of sediment, and then yeah, you can. There are various techniques been used for kind of upscaling the carbon production. And so Joe Cook had a really nice paper on this some years ago, trying to upscale across Greenland, and we've looked at doing that for some of the Antarctic stuff. One thing I'm not really sure is how useful that is, but that's something that I think the Black and Bloom project kind of tackled, and trying, figuring out how to incorporate those measurements into models. So I'd say that would be something that I would leave to them. But yeah, I try and do it per gram of sediment because that's what is basically in the literature at the moment.
Leanne, nice to see you. Hello.
Hi, thank you, Liz, for a fantastic talk, and really nice to see how that worked out. You showed some really nice data from EFA on the oxygen profiles and going anaerobic. And then you said in other systems, you form these balls. So if you have all of a sudden a structured cryoconite where you don't yet have the balls, where you have anaerobic conditions underneath and then aerobic at the top, in the moment you flush them, what happens? Do you form balls with the structures anaerobic inside, aerobic outside? Because I think Takeo Nozumo has shown something like that in the past. And once they deposit again somewhere, do you again form this layering? That means, is it like a lake? That means you turn them over in a way, you make them, bring them out, bring them back in. So is that the kind of process that you envisage with because you showed three systems where you have like stable cryoconites, floating away cryoconites, and so because they're actually dynamic systems. So how do you envisage that happening?
So I think the lake turnover is a really nice analogy there, and I think that happens. And whatever showed with disturbing the system and then doing the profile every few minutes shows that yeah, you disturb it and then within a couple of hours, yeah, those anoxic layers form. But I think also when you disturb them, the little granules do persist when you disturb them. And yeah, if you profile across those granules, and Nozomi's done that, and Eva's done that with some granules as well. If you get the needle right through the granule, then you see an anoxic zone in the middle. So I think you get the lake turnover of the whole cryoconite, but then I think some of those granules, if they're pretty hard to squash, if you've tried to kind of break up a granule, they're pretty solid things. So I think those persist. So you still get those anoxic zones stay there, but then the larger anoxic zones within the kind of cryoconite layers do get turned over and reoxygenated, I think.
So just a follow-up question on that. In that moment, basically, and I know you said I'm not asking about microbiology, and I'm sorry I'm going there, but basically what means that the microbial communities which produce that anoxic environment, they sort of like become food, that's your meals on wheels, and then some of them must survive because they reestablish themselves again. So are you saying that the ones which survive inside the granules and then sort of say, ooh, okay, I'm back and continue?
I think, yeah.
Because you explain the chemical process, but I'm trying to think from the point of view of a bug.
I think a lot of the anaerobes that Eva's identified are not actually true anaerobes. They're facultatively, that's hard to say, facultatively anaerobic. So I think if you take them, if you put them in an aerobic environment, it's not like they just die. So I think there's a few true anaerobes, but not many. Would be kind of my understanding of it.
Thank you.
Charles, go ahead with your question.
Yeah, hi Liz, thanks a lot for a really great talk. I had a question for you. You showed the great diversity of these cryoconite holes and the different shapes and the different light regimes. So it got me wondering, can you create these things in the laboratory to do controlled experiments? So I was just thinking as I was watching it, could you have a block of ice, for example, with some dark dust on the top with a light above it, and create cryoconites in the laboratory where the conditions are much more controlled, and then you could manipulate communities and add different organisms? Is that something that you thought about, or that's even practical?
So Jazz Miller is doing that right now as we speak. So we've messed about with doing this for a number of years. The difficulty, particularly if you're trying to grow an ice-lidded one, growing an open hole is relatively easy. And I know Arwen's group in Aberystwyth have grown some beautiful ones. But trying to get the ice lid to grow over them without freezing the whole lot, and then getting them to melt again without ending up with a puddle of water is a bit of a pain, but you can do it. The tips are use deionized water, boil it 3 times, because then you get the right kind of bubble formation happening. And what Jazz is trying at the moment is we've bought some plastic segregated boxes, and because they're trying to obviously get some statistical power on this, so doing replicates as well is a right pain, because you can grow one beautifully and you can do all sorts of things to it, but to actually figure out if what you're doing is a trend or just by chance is much harder. So what Jazz is doing right now is hopefully not making puddles of water. They sent me some photos yesterday, and it looks like, yeah, they're growing really nicely. So Jazz is doing that to make Snowball Earth cryoconite. But yeah, one of the things that we'd love to do would be, seed them with individual microbes, figure out who are the ones who are actually important to make these systems work. But yeah, we haven't quite got there yet.
Thanks.
Anyone else for a quick question? John, hiya.
Hi, Liz. Great talk. Thanks very much. So I'm just wondering for your thoughts about, you mentioned that possibly cryoconites could serve as an analog for icy satellites. So if you had an orbiter around Europa, based on what you know about cryoconites, what would that orbiter look for?
Wow.
I mean, in terms of, is there a biosignature from the cryoconite that can be sort of uniquely, diagnostic of that type of community from orbit, potentially, in a remote sensing approach or something?
I don't personally, I don't know, but one thing that you could look at would be the organic carbon. I think again the Black and Bloom project might have done some cool stuff on trying to quantify cryoconite from above. And one of the things that the Bristol group has also done, Alex Anicio did in the past, was looking at how cryoconite darken over time as you get this production of the organic carbon. So you could try and look at the color of any debris that you see and figure out if it was organic. But yeah,
Do you
I don't
reckon
know.
there might be any sort of diagnostic sort of absorption feature or biomolecular stretch from a chlorophyll or some type of a pigment or anything like that?
Didn't Black and Bloom also look at spectral, trying to use satellites to look for those like green chlorophylls, red chlorophylls?
Yeah,
a vague memory of that.
yeah, I think that would be the kind of closest we get. I just want to get a lander in there and measure it. But yeah, we're probably a few years away from that. Yeah.
Thanks.
Yeah. Okay. So I certainly have to shoot off, but it's been brilliant hearing from you, Liz, and amazing seeing your face after so long. It's always good to see. Just a quick message that in 2 weeks' time, we will have Olga Golishina from Bangor University talking about acidophiles and cold temperature archaea. So we would love for you to join us for that again, and have a good week or two in the meantime.